Hello, everyone, and welcome to the Wild Neighbors Speaker Series. We're very happy to have you here today. This webinar series is a collaboration with Travis County and the City of Austin, who co-manages the Balcones Canyonlands Preserve, along with a number of other private and public partners. Before we get started today, I want to introduce myself. My name is Jeremy Hull, and I'm the Community Liaison for Travis County Natural Resources. Today, we're very happy to have Ruben Tovar, a PhD researcher at the University of Texas here to talk with us about the diversity and evolution of the Eurisia salamanders that we have in Texas. After the presentation, Jaya Torres with the City of Austin is going to manage a Q&A session. So please feel free to put all your questions that you may have during the presentation in the Q&A box, not the chat, and we'll get to as many of those as possible at the end. We are recording this webinar and it will be posted to Facebook and YouTube to be used as a future resource. And on that note, I'm going to stop sharing my screen and hand it over to Ruben.
All right, thank you, Jeremy. Okay. Well, good afternoon, everybody. My name is Ruben Tovar. I am a PhD candidate at the University of Texas at Austin in the Department of Integrative Biology. Today, I'd like to talk about exploring the diversity and evolution of the Central Texas Eurisia salamander clade. But before I get there, I'd first also like to thank the organizers of this Wild Neighbors series. Jeremy and Jaya have been incredible in helping me to prepare for this. So I really appreciate it. I really appreciate the invitation. And so here I have depicted a couple of salamanders, but even before we get there, I think I'd like to take a moment, just a quick moment to recognize that this month we're still in the month of Hispanic Heritage Month. And so I'd like to just kind of point out a couple of people that have been pivotal in my academic career as I've come to my PhD or made my way to my PhD. The first person I'd like to recognize is Dr. Dana Garcia. She's a professor of biology at Texas State University, and she is my former advisor during my master's program and really helped me to organize my thoughts and questions regarding some of the research you're going to actually hear today, and you'll see her again here in a bit. So thank you to Dr. Garcia. The next person I'd like to recognize is Dr. Yolanda Flores-Niemann. She is a former professor of psychology and interim chair at the University of North Texas. She's now retired, but she's written a number of books on dedicated most of her research understanding how minorities in academics have had quite an uphill battle, and has dedicated her life to trying to change that. So most people in the professional world know her as Dr. Yolanda Flores-Niemann. I have the privilege of knowing her as Aunt Yolanda. And if it weren't for these two Hispanic women in academics, it would have been a very, very tough road for me reaching this place. Okay, so back to the salamanders. So I mentioned that I'd like to speak a little bit about the diversity and evolution of Texas Eurisia. And so I'd like to kind of break this whole talk down into two major sections. The first, Texas Eurisia diversity, and we'll think about species identification, how biologists do this, and some lessons that we've learned from morphology and genetics. The next section I'd like to think about or speak about is the Texas Eurisia evolution, and in particular, evolution or evolving to life in perpetual darkness, as some of these or a handful of these species have done. So let's start with Texas Eurisia diversity and species identification. So let me introduce the Texas Eurisia really quick. This is a group of salamanders. It's composed of about 14 species, and that's been increasing slightly here just recently, actually. They're endemic to Central Texas, and in particular, they're endemic to the Edwards Plateau and its associated aqueous habitats. So the Edwards Plateau is this unique geologic uplifting composed of the carbonate rock limestone. And after millions of years of erosion via dissolution, which is this weakly acidic rain, it eats away at the limestone, creating a number of microhabitats all along the Edwards Plateau. And so broadly speaking, we can categorize these habitats into two, or at least I like to think of them as two fundamental components. So one might be a surface habitat like Honey Creek Springs depicted here. And if we think about the Edwards Plateau and we think about where we see springs, they align the edge of the uplifting, where you get the release of the aquifer from geologic shifting. So here is an example of a spring habitat and what I would associate as being a surface habitat. On the other hand, there are subterranean habitats as depicted here by this cave where I am looking up. I'm usually looking down, but just for this picture, I was kind of doing a little pose here. But yes, salamanders are usually down in water. But this is depicting the other kind of dichotomy within this, within the potential habitats that these salamanders occupy. One's a surface and the other is subterranean. So it might not be, super surprising that we find salamanders associated with these habitats, and they look quite different. And so I'd like to think about this a little bit more as we move along here. In the surface habitats, we have salamanders. This is Eurecia latitans, or the Cascade Cavern salamander, and it has pigmentation and relatively well-developed eyes. Where in the cave and also in other subterranean habitats, we have these subterranean characteristics like reduced pigmentation and eyes. So before we get too far into naming out these characteristics presumably associated with these habitats that these salamanders have evolved in, I'd like to introduce a little bit about what makes the Central Texas Eurecia different than maybe your typical amphibian. So let's start with the typical amphibian life history. Well, that usually includes two major life history events. So one is that once a salamander hatches from the egg, it's a larva. And at the larval stage, most amphibians maintain a larval stage in the water in some kind of aquatic environment. Eventually, they go through metamorphosis. They lose their gills, lose their tail fin. They have other things that change around their body, and they eventually become terrestrial. So how are these salamanders different? Well, the salamanders here in Central Texas, or at least the group that I'm going to be speaking about today, are considered neotenic. That is that they retain a juvenile state or larval state while at the same time reaching sexual maturity. So what does this mean? This means that the salamanders here in Central Texas never leave the water. And here, depicted here, is a Jollyville Plateau salamander, Eurecia tonkawa. And you can see that it has this little fluffy gills on the side of its head. And you can't really see from this angle, but it also does have a bit of a tail fin on the side. Okay, well, that's pretty neat. So they never leave the water. They spend their whole life in the water. And so that means that they are reliant on the pristine water of the aquifer that underlies them. Okay, so I now would like to dive into a little bit about the diversity within Central Texas. So how do we measure diversity? Well, biodiversity is very simply put, measured by way of counting the number of species that we can identify. Okay, Well, historically, how we've done this and how biologists have done this, that is species identification, is to identify characteristics of the organism that set them apart from other organisms, but are still maintained within their population or within their species complex or group. And so, so that's all fine. And in fact, we've talked a little bit about how surface characteristics and or morphology are commonly seen within these salamander species. And again, they include pigmentation, relatively well-developed eyes, and these kind of small, robust limbs. And the same is true for subterranean species. They have reduced, or the same is true is in that they have similar characteristics to one another, in that they have reduced eyes, pigmentation, and elongated limbs. So when this happens, this is a phenomena in evolutionary biology called convergent evolution. And when your only measurement for identifying species is the external characteristics and morphology, well, this can be kind of misleading, because you can imagine how one might accidentally group two different species that look very similar together, because you might find a species at cave 1 over here and a species at cave 2 over there, but they're both evolving to living in perpetual darkness. And so they both have similar characteristics, and they both converge on these morphologies. And so this has historically been problematic. So allow me to introduce the way that we measure, another way we measure species identification, and that is through phylogenetics. And phylogenetics is a way to measure the relationship of species based on their DNA. And to your left here is a phylogenetic tree published by Devitt et al. In 2019, and is the most recent and comprehensive phylogenetic tree of the Central Texas Euresia. And so let's for a minute walk through another scenario here. So let me reintroduce again Honey Creek Spring and Honey Creek Cave, and I just put these same pictures up here. And at Honey Creek Springs, we have the salamander that looks like a surface salamander, and at Honey Creek Cave, we have a salamander that looks like a subterranean salamander. Well, if we were maybe just going purely on morphology, I would definitely call these two different species. But what we have found is that they are in fact the same species, and they're actually part of the same population, which is pretty cool. So you get this dichotomy you get this dichotomy, two different looking salamanders that are the same species. That wasn't completely clear until we actually introduced genetics into the picture. The other usefulness about using genetics in identifying species is that oftentimes we get to identify species that are completely novel to science. And so here are two examples of such species. And again, they're subterranean. Okay, great. So I'd like to walk everybody through. Again, this is based on the Devitt et al. Publication in 2019. I'd like to think about what species we have recognized using this genetic approach. And so I'm just going to kind of walk through this map, this distribution map, and show you some pictures and some of the diversity of the Central Texas Euresia salamanders that we have identified. So let me start with the Western group. And historically, this Western group, until this publication, was all known as one species. And so that was Euresia troglodytes, or the Valdina farm salamander. Since this publication and the genetic analysis associated with it, Devitt et al. has proposed that, in fact, there are two other species within this complex, which was quite phenomenal. And we are working, and Tom Devitt is working on disentangling these species. Okay, so we'll move now to the this other subclade called the Eastern Blepsomolgi. And here we have Euresia terophylla, Euresia neotnes, and Euresia latitans. Another very interesting subclade of the Central Texas Euresia is a subclade called Typhimulgi. And Typhimulgi, all the species that reside in it, are associated with very deep portions of the aquifer. And so they include the Texas blind salamander, Euresia rathbuni, seen here, the Austin blind salamander, Euresia waterloensis, and this really weird individual, Euresia robusta, which has in and of itself its own story. This is the only specimen that we have of this species, and it was collected in 1951. And so we're currently trying to extract DNA using several methods, including ancient DNA methods. Here is me and Dr. Anat Bellison. She is a postdoctoral fellow in Dr. Kelly Zamudio's lab here at the University of Texas. And so her and I have embarked on trying to extract DNA from this one very old sample. We will see where that goes. Okay, and so north of the Colorado River, maybe where a majority of people here are interested in, are these three species, and I'm going to show you them now. Let's see, Euresia chiselmensis, or the Salado salamander, the Georgetown salamander, Euresia nephragia, and the Jollyville Plateau salamander, Euresia tonkawa. And I have two individuals here, and they're both from different localities of Euresia tonkawa. Because Euresia tonkawa is relatively widespread, I decided to go ahead and include these two individuals. What's really cool, though, and relatively recently found, is a new species to science, and that is unnamed yet, and we are currently exploring it. And it seems to be the only subterranean species north of the Colorado River. So we're very excited about that. Okay, so I've spoke a little bit about the Texas Euresia diversity, a bit about how the neotenic life history state means that the Euresia salamanders here in Central Texas rely on the pristine aquifer water that we also drink and rely on, and how convergent characteristics and morphology have added challenges to identifying species, and how we have attempted to disentangle that using DNA and molecular approaches. So now I'd like to think a little bit about about Euresia evolution, and in particular, again, thinking about life in perpetual darkness. And so for the most part, my dissertation is focused on thinking about this question and thinking about convergent evolution in subterranean species through development. So let's consider a subterranean species. This is a Texas blind salamander, Euresia rathbuni. And let's consider some of the sensory modalities or characteristics that you might think are associated with something that has been evolving to live in perpetual and thrive in perpetual darkness. And so for the most part, the way I like to do this in my head is think about it as two major groups. Think about them as two major groups. One is thinking about expanded characteristics, things that might be heightened. And the other is to think about other senses or characteristics that might be reduced, because perhaps you just don't need them when you live in perpetual darkness. Okay, so characteristics that might be expanded may include, or at least with the salamanders have been identified. They include a broad head, a heightened sense of chemosensory, heightened pressure sensory system, otherwise known as a lateral line, and these elongated limbs. Okay, so what about reduced things, reduced sensory system modalities or characteristics? Well, maybe a slower metabolism. They certainly have less pigmentation by way of melanin, and probably a reduced circadian rhythm. But for the purposes of my research and the future, the way that I like to think about these organisms a little bit more is to think about this idea, what we can't see pretty much, which is the reduction of their eyes. And so it turns out that we were lucky enough to be funded by the NSF to really dig into this question and understand the developmental and genetic pathways associated with convergence of these reduced eyes and other subterranean characteristics associated with these some of these species. And so some of my past work during my master's included very fundamental approach of describing histology, because none of this had been done yet. And so here is Eurecia sessorum, or the Barton Springs salamander, and here is an adult eye of Eurecia sessorum, and you can see it has a well-developed retina and a lens. So a lot of the gross anatomy that one might expect in a sighted animal. The same is true for Eurecia nana, or the San Marcos salamander. It also has a well-developed retina, and it's not depicted in this picture, but they do have a lens and an optic nerve, and all of these other gross anatomical structures that we might associate with an organism that has vision. On the other hand, the Texas blind salamander had quite the opposite. It had underdeveloped retinal layers, did not have a lens, it had a bit of an optic nerve, but we don't know how far reaching that was. And all of this underdeveloped eye was completely surrounded by pigment. So it was quite bizarre. So histology allowed us to kind of make some of these observations, and they're fundamental to future questions. However, performing histology on organisms is quite destructive, although it tells us a lot. And so we were interested in maybe thinking of a technique that would be a little bit less destructive, but also give us an idea about the gross anatomy, ocular anatomy of these salamanders. So to do that, we used this relatively new technique called diffusible iodine contrast enhanced computer tomography. And I know it's a mouthful, so I'm just going to refer to it as DICE-CT. The way that DICE-CT works is that it's a micro CT scan, and for the most part, micro CT scans will pick up on dense material. And that's not very helpful when you're interested in soft tissue like the eye. So what we do is we stain the specimen with iodine overnight and then micro CT scan it. And what that does is the soft tissue absorbs the iodine stain, and this renders the soft tissue visible in the micro CT scan. So it's a very, very cool technique. All right, I'll let it play out here. After we receive these scans back, we can then put them into software that recompiles and consolidates all of the different sections all together, resulting in a 3D image like this. So this is a 3D image of the Jollyville Plateau salamander. This is the same individual that you saw earlier in the distribution map, Eurecia tonkawa from Testudo tube. And so we can get this image, and it allows us to also cut through all the soft tissue layers and identify all of the gross anatomy that we might be interested in. And so here I have it rotating, and eventually you'll see it cut away down through the head. And you can see all the soft tissue, the muscle, the bone, the brain, the inner ears, and of course, what I'm interested in, which is the eye. Let me pause it at the end there. And there you can see the eye and some of the retinal structures in back of it. So we've done this for a number of species, adult species here. Here is Eurecia raphini, the Texas blind salamander. This is the exact individual that you see pictured on the right-hand side. What's really cool and fascinating about this DICT scan as well is that it allows us to also identify features of the lateral line. So I don't know if y'all can see it out there, but from my view, you can see all of these little bumps, and let me see if I can point them out. All these little bumps here, or dots, along the face. These are all what are called neuromass. And neuromass are pressure sensory cells all along the skin of the salamander that allows it to sense pressure differences in the water. There is a graduate student at Texas State now working on quantifying these neuromass between surface and subterranean salamanders. Here's an example of the San Marcos salamander, Eurecia nana. And again, you can see all of these little dots and their distribution along its head. Neuromass. Okay, so I said I was interested in development though, right? So I've introduced these desperate looking salamanders, subterranean and surface. And so what are our predictions when it comes to underdevelopment of the eye? Well, here's our working hypothesis and how I might think about eye divergence. So from left to right, what we have is during development, early development, we predict that similar eye development is happening. And at some point, as these salamanders continue to develop, they start to diverge. One salamander that is, say, surface species will continue to develop an eye, while, say, a subterranean species might have cease of development of the eye, and maybe even active degeneration of the eye. So we've been able to identify where this kind of commonplace, this starting point of commonality between different salamander species occurs. So here's an example of the Cascade Cavern salamander, Eurecia latitans, about 1 month post oviposition. And this is right about hatching for all of these salamanders. So we can also do the same thing that we did with the adults. So we've DICT scanned these salamanders. We can render them in 3D software, and then we can cut through and identify anatomy of interest. Of course, here being the theme, we are trying to look for similar eye structures. And you can kind of see it from here. You can see a lens and the retina developing. We can then go through the software and color in the anatomy of interest. So here is a colored in version of what you just saw. And so, you know, my kids get their coloring book at home and I get to color in the 3D software. So everybody's happy. But what's really cool about this particular method is that we can consider all of the eye and not just a 2D image. So we're able to quantify the whole optic cup and all of the lens. And the way that we do that is we take the volume. So we're able to do volumetric analysis using this technique. Okay, so we have been, or I have been working on filling in all of these developmental stages. And so here at the very end is that stage 1 month post oviposition. And we have continued to increase our staging for both blind and sighted salamanders. And of course, we've also been collecting adult salamanders as well. So what does this tell us? Well, it turns out that some of the fundamental, both molecular and developmental underpinnings that are responsible for eye development are conserved among all organisms. And that includes humans as well. And so when you have a naturally occurring system like blind salamanders here in Central Texas, it really gives us an opportunity to understand what molecular mechanisms are underlying the underdevelopment of an eye. And so a close parallel to what we see in humans is usually something that goes awry during our development. So for example, this congenital disorder called aniridia has historically been identified as the underdevelopment of the iris, which is the colored part of your eye. And to your right here, you can see a healthy human eye on top, and then a person with aniridia on the bottom. But importantly, aniridia is diagnosed as a panocular congenital disorder. So you can have any number of these eye disorders listed here. So, and some are relatively common, actually. So cataracts, macular and nerve hypoplasia. So it's not just about the development of the iris, but it also extends into other aspects of eye disease. And so again, this group of salamanders here really allows us a window because we can compare a salamander with a relatively well-developed eye and a salamander with an underdeveloped eye, really allows us to disentangle what might be happening at the developmental and molecular level when it comes to underdevelopment of the eye. Okay. So Texas Eurecia evolution, the Texas Eurecia is an ideal model to understand all of these really fascinating, interesting questions regarding evolutionary development, including convergence and parallelism. But they also might be a potential model to understanding human congenital eye disorders as well. And so I'd like to kind of leave you a little bit with this thought too. So this is a relatively recent posting on social media about Jacob's well, and that, you know, I'm sure y'all have heard this before, but salamanders that rely on the pristine water of the aquifer, Edwards and Trinity Aquifer, have often been identified as indicator species or canaries in the coal mine or barometers for the Edwards and Trinity Aquifer, which are all completely correct. And they're some of the first to be affected by bad water quality. And so, you know, as we move forward, we really need to be conscious about, you know, how we use water and what that might mean to the organisms that live in that water as well. Because when we think about it, this habitat both adds to our livability and is a resource that we rely on as humans. And if that's not convincing enough, then, you know, I would really consider the fact that we have only scratched the surface when it comes to what these salamanders might be able to show us when it comes to underdeveloped eyes and how that might link to human medicine and congenital disease. So with that, I'd like to thank the Hillis Zamudio Canatella Lab, Dr. Tom Devitt, Dr. Dana Garcia, the NSF, and our collaborators at the San Marcos Aquatic Resource Center, the U.S. Fish and Wildlife, also the University of Texas High Resolution X-ray Computer Tomography facility here. And I'd love to take questions.
Awesome. Thank you, Ruben. Those micro CT scans that you showed are just so fascinating. I could watch that one loop for hours, I think, and watch it go back and forth. But as you said, we are going to be doing a question and answer session. We already have a few in the Q&A box, so we're going to start getting to those. But if you have any other questions for Ruben, just start populating the Q&A box and we'll get to as many as we can. I'm also going to post several links into the chat for anyone that wants to find out more about the history of the BCP, how we were formed, and events that we have going on. With that, I'm going to turn it over to Jaya for the Q&A.
Great, thanks. Ruben, that was a great presentation. We're going to go ahead and dive into the Q&A. So first question was earlier on in the presentation, came earlier on in the presentation, and it was, what Texas counties are these salamanders in?
Oh, geez. Bexar, Comal, Travis, Williamson. What county is Uvalde in? Kendall. Geez, I'm missing some. I know I'm missing some, but those are the ones off the top of my head.
Great, thanks. Next question. Did the lateral lines evolve independently in salamanders?
Independently from, I'm guessing, fishes?
Right.
Relative to fishes? No, I don't. Well, I don't know. So I don't know that anybody has really disentangled that question. I don't think so, because they're very similar in structure, and thinking about the most common ancestor between fishes and salamanders, which is a long time ago. I think I would say that they're probably features and characteristics of the lateral line in salamanders that are different than fishes, but because of their own kind of evolutionary trajectory. But if we look deep enough, I'm pretty sure they all, for example, the lateral line in zebrafish and in salamanders is known to come from a placoid early in development that migrates from the ear, and it's a combination of the inner ear placoids and the hindbrain placoids that migrate down the body of the organism. So in that way, they're still the same, but each is on its own evolutionary trajectory. So I would imagine that placement or the amount could potentially be different. So there would be at some point some divergence, is my prediction, in the lateral line, but I think they are coming from a similar place, evolutionarily speaking. So for example, sharks have like a pretty heightened lateral line, in that they've even taken it a step further and kind of beefed up their ampullary organs. Ampullary organs I didn't talk about here, but they are part of the lateral line, just like the neuromass, and they are electromagnetic organs, and so they can sense electromagnetic fields around them. Other fishes, some bony fishes, have weakly electromagnetic heightened ampullary organs as well, because they produce weakly electric fields. So yeah, I think the lateral line is quite diverse, but it all stems from a similar evolutionary origin.
Awesome. All right, next question. What is chemosensory?
Oh, great question. Sure, yeah. So chemosensory is, simply put, the sense of smell. Yeah.
Okay, makes sense. Next question. Aside from the Cascade Cavern salamander, are there any other species or very closely related species with both surface and subterranean morphology?
Right. So yes. So Eurecia pterophila would be another example of what we call a polymorphic species. Excuse me. So that's a fancy term for saying that we find both subterranean and surface phenotypes or populations of this species. So the answer is yes.
Okay, and kind of similar topic for the next question is, do all subterranean species have an above ground form?
No. No. I mean, so I guess it's easy to, so let me elaborate a little bit more on that. So what's interesting is that if it's almost like every spring kind of, not every, but a lot of springs have their own endemic population of surface salamanders. And usually what you find is that throughout history, like geologic history, what's happened, or what we think has happened that has led to a number of species that have these subterranean phenotypes, is that there have been a number of invasions into subterranean habitats by these surface species. So what's happened is that somewhere in geologic history here, we had an invasion of a surface salamander into the subterranean habitats, and they continue to evolve over millions of years and have resulted not only in its own unique species, but also in these crazy looking characteristics and phenotypes and morphologies that we see. So they're not related anymore, right? They're different species. But oftentimes, for example, in San Marcos, there is the San Marcos salamander at Diversion Springs and Spring Lake. But then below that, way below that, is the Texas blind salamander, right? Two completely different species, but they just one happens to live on top of the other.
Okay, great, thanks. All right, this next question is a little longer, but it's really interesting. So is the large diversity of neotenic salamanders here due solely to the abundance of springs and aquifers, or would the drier environment of the hill country provide an environmental pressure to discourage completion of the typical life cycle?
That's a wonderful question. Yeah, we would love to know the answer to that question too. It would be really cool. So that is certainly one hypothesis. One hypothesis is that over, again, geologic time, a long time ago, there had been major drought events. And there is at least one paper that I can think of that comes to mind where scientists have tried to identify these major events using stalactites in caves and using a kind of carbon dating type of approach to understand this. And it does seem, this was a paper published in Science, oh gosh, not too long ago. And it did suggest that there have been major drought events here in Central Texas. And so that is certainly one hypothesis, is that why leave the water when you already have a great resource just below you, and things are, drying up really quickly up on the surface. And so it's also unclear how this has contributed to migration events underground. You can imagine a scenario where you have a major geologic drought like that, and springs dry up, rivers dry up, and the only thing that's not dry is underneath where we can't see, right, in the Edwards Trinity Aquifer pools. And they retreat down and follow the water, and they just stay in the water. So that is certainly a hypothesis for both why they would maintain a juvenile state, a neotenic state, and a hypothesis for how some of these subterranean species might have just kind of hung out down there and remained in the subterranean regions of the aquifers. That's a great question. Yeah.
Awesome. Yeah, it's a good question. All right, next one. Is there a good place to observe salamanders in the Austin area?
Sure. Well, let me take that back. So Austin area. So most of these salamanders are threatened and or endangered. So you can observe them. So I want to say, I think, so at Barton Springs, there are some in tanks. I don't think you can go to places where, at least in Barton Springs comes to mind immediately. and you can observe them there. When it comes to in the wild, I think a lot of these places are kind of protected, and the salamanders themselves are also protected, just because they're fragile and threatened. So I would say maybe, if you're okay with observing them at, in a tank and controlled environment type of scenario, that maybe heading down to Barton Springs would be a good idea.
Great. The next one, U.S. Fish and Wildlife recently found that E. robusta is likely not a valid species. Is this something that your lab is studying?
So yeah, they did, and they asked us, and all we have is morphology, right? All we have is one individual, and so so Dr. Bellison, who I am shadowing using this ancient DNA protocol, was doing this for a number of museum specimens, and so I just kind of jumped on the bandwagon, and I'm hoping that we can be successful in extracting DNA, because we just don't know based on the one individual and morphology. And so if we can have some genetic data that would allow us to include it into a phylogeny, that would be extremely useful for everyone, us and the U.S. Fish and Wildlife as well. So, yeah.
Okay, great. And in reference to the previous question, someone pointed out that the Meadow Center at Spring Lake in San Marcos has the San Marcos salamander and the Texas blind salamander in tanks.
Yep.
And then that there's also a glass bottom boat tour where you can observe them in their habitat. So
yes.
Point that out too.
Yeah. I didn't know what around Austin meant. If you're willing to drive, yeah. If I-35 is kind to you, it's not kind to me usually, but sure, and it's about an hour or so drive to San Marcos.
Right.
And I'll give you one other little tidbit about the Meadow Center too, and the Rathbuni that are there, the Texas blind salamander, at least a couple of them. Back when I was doing my master's with Dr. Garcia, I worked with the U.S. Fish and Wildlife in San Marcos, and I bred their first F3s for my research, and a couple of them ended up making it to the Meadow Center. So those were some of the offspring that I had privilege of reproducing back in the day.
That's very cool. I like that.
Yeah.
It's a good connection.
Yeah.
All right. So next question. You mentioned the Valdina Farms salamander. Is that extinct, or is it considered the same species as others now?
Nope, it's not extinct. It is its own standalone species. Yeah. So what Dr. Devitt found in his analysis of the genetics is not that the Valdina farm salamander is completely gone, but rather what we thought were the Valdina farm salamanders in other areas of West Texas turned out to be their own species, or seemed to be their own species based on the genetics. So the Valdina farm salamander is still very much alive and well in a number of localities, including like Lost Maples. Okay.
And I know this next question, you've kind of touched and mentioned, that most of these are threatened or endangered. Which ones are listed as endangered?
All right. So the Texas blind salamander is endangered. The San Marcos salamander is endangered. The Barton Springs salamander, the Austin blind salamander are all endangered. And threatened include all three species north of the Colorado River. So the Salado salamander, the Georgetown salamander, and the Jollyville Plateau salamander. And I should note that when I'm thinking about this, I'm thinking at the federal level as well, right? So as we all know, there's a federal category, and then there's a state category. And so at the state level, I think it's even more inclusive when it comes to salamanders listed as threatened.
Okay, great, thank you. This next question is kind of going back towards morphology, but why did they evolve with external gills as opposed to internal? Or
That's a good question. Yeah, I think it's that's a good question. Again, I think this is something that we have to kind of backtrack all the way to when salamanders first started evolving. And so as far as I know, it's just they have always had gills at the larval state. So yeah, that's a great question. I don't know that we know why they had external versus internal. It probably has to do with the craniofacial development of salamanders, broadly speaking, salamanders everywhere type of thing, and also include all of the extinct species as well that were lineages of the extant current species we have today. That's a great question.
Okay, thanks. Next question. Is the endangered status due to habitat loss, small localized population to begin with, or a combination of both?
Both for sure, yeah. As I mentioned, and I didn't really elaborate on here, but a lot of these, a number of these species are only known from only a handful of localities. For example, the Texas blind salamander, we know it from about 5 localities or so, and so that's not very many. The San Marcos salamander is only known from Spring Lake area. The Austin blind salamander only from Eliza Springs and Old Mill Springs. So the localities are, there's not very many places that these salamanders were able to observe and actually see these salamanders. And so I agree that the other thing about that is that, yeah, that the habitat itself, our water usage is also, given our growing population in Central Texas, is something of concern. So.
Okay. And then one last question. Do the Eurecia we see in the area migrate or move between different springs?
Oh, great question. Yeah, that's a wonderful question. We're trying to figure that out. It seems like they do. And so yeah, so at that point, you start to get into a subdiscipline of biology known as population, or use different techniques. What's called population genetics. And so in order to disentangle that, you need a large sample size, several localities, and some hypothesis that you think that some migration might be happening. But absolutely, like it's an absolute possibility, and based on some very preliminary results in a collaboration I'm involved in, it seems like they can migrate. Yes. I mean, and it's also kind of, we say migrate, right? But it's for the most part, there's probably a lot of interaction between the hydrology, the geology, and the biology, mainly the behavior of the salamanders, that all come into play when we think about the word migrate, right? So when it's a drought, what kind of corridors, hydrologic corridors, does that open or close? When it rains and floods, what kind of hydrologic corridors does that open and close? And how often does that happen? And how much do we see that in the genetic analysis that we're able to do at the population level? But yeah, it's a really neat question. Yeah.
All right, great. Well, we had really good questions, a really great presentation. I guess if you would like to share your email in the chat if folks have other questions, but don't feel obligated.
Oh, shoot. I meant to put it in my slide. I'm sorry.
Oh, that's okay.
Let me put that in here.
But yeah, I think that is all the questions we've got, and we'll go ahead and wrap it up now. And yes, everybody should be able to see that email address there in the chat.
Oops. Okay, great. Well, thank you guys.
Yeah, thank you for joining us, Ruben. That was awesome. Everybody have a great day, and thanks for joining the Wild Neighbors.